Okay, thanks, Federico. So I'm going to take care of presenting the second, the task 2, the ideation challenge, which didn't involve any coding, but I hope it's going to be interesting anyway to see. So it prompted us to think about ways in which we could exploit DPEG's cohort data to better understand the etiology of hypercholesterolemia beyond what's already known to be a risk factor. And the approach that we proposed in task 2 is centered around gene-environment interactions, the discovery of these. Because of that, I would like to briefly mention a work that actually motivated us to take this direction. And this is an unpublished work from a colleague of mine, Saul Pierotti at the European Bioinformatics Institute. And very briefly speaking, they run genome-wide association studies on Medaka fishes, these small Japanese fish that can be inbred directly from the wild. So they generated F2 crosses of between several inbred lines, and they tested the associations between genetics and quantitative trait, which is the heart rate in embryos. But they did so varying the environmental conditions, and specifically the temperature. So you can see that at 21 degrees Celsius, the reference homozygous, the heterozygous, and the alternate homozygous have basically the same heart rate. So this particular locus that is plotted here wouldn't have been discovered as a quantitative trait locus. But at 28 degrees Celsius, the alternative homozygous has a lower heart rate with respect to the other two, and this effect is even more pronounced at 35 degrees Celsius. These are temperatures that are in natural range. So there is a clear evidence of interaction between gene and environment, and they observed some degree of these interactions in 11 out of the 16 loci that they discovered. So if we can learn something from this, is that the presence of gene-environment interaction seems to be rather prevalent, and they also observed that the variance explained by the interaction effect is in many cases very high and even higher than the usual standard additive gene effect. And because of that, in some cases at least, the contribution of the genetics to the trait cannot really be detected if not stratifying for the environment. And so we wondered what happens in humans actually. And for the task 2, we had to devise a way to account for the environment in humans without, though, breaking ethics and exposing thousands of people to different temperatures like Medaka fishes or chemicals or sleep patterns and so on. So the standard way of testing associations between genetics and environment is with this linear model that Federico already showed before, where you have a genotype value and an error term and an intercept for quantitative traits. And so here you're simply assuming that there is a causal path between the genetics and the outcome Y. If we want to also integrate the environmental variables, we can add more term to this linear model, and we can have an environmental variable with its own estimate of effect, coefficient beta 2, and also a term that accounts for the interactions between the genetics and the environment. And so what we are assuming is that the environment can have an effect on the outcome Y that is independent from the genetics, but then we are also allowing a pseudo-exposure X that mediates both the effect of the genetics and the environment, and this is where the interaction can happen. So any effect that is not captured by the estimates beta 1 and beta 2, according to this framework, has to be an interaction effect. But the reality is that in human, even though this is pretty straightforward, in human population genetics, this is not really used. interactions are difficult to understand and uncover. And this is because typically there is the genotype and the interaction term have a high collinearity, and the estimates of their effects will covariate, and this translates into higher uncertainty of these estimates. And in the end, you have low power in testing whether the interaction effect is actually there, is different, significantly different from zero. And so, while addressing task 2, we found this work, which was published this year, where Zhu and colleagues proposed a rather elegant solution to this problem, and to increase the power for this testing. And so looking at the 2 equations that I showed before, they showed that the genetic effect in the absence of the environment, the estimate of the genetic effect in the absence of the environment, and the estimate of the gene effect in the presence of the environment, if there is no interaction between gene and environment, they will linearly correlate. And this means that we can look systematically for points, beta 1 and alpha, that deviate from this regression line. And so what we proposed for task 2 is in the PEGS cohort, which has a very rich characterization both of molecular variables and environmental variables, to systematically fit these linear models to genotypes and environments variables, and then obtain the estimates of these coefficients, model their correlation in the hypothesis of no interaction, and eventually test the deviation of these points from this expected correlation. And this gives us a list of prioritized gene-environment interactions that we can assess forward. And this can be very interesting because in a way we can inject the knowledge that we have of the environmental variables into the mechanistic interpretation of the genetic variants. So I'll show a quick toy example about this concept. Assume that we have a genetic variant that is sitting on an enhancer that is poorly annotated. We may assume that it has some regulatory function, but we really don't know what it does. And we discover an interaction with an environmental factor that we know the exposure to this environmental factor as the effect of increasing the concentration of a certain metabolite. So if we look in the neighborhood of this enhancer, we may find several genes that plausibly could be regulated by this enhancer, and each one of them has a protein product, for example, A, B, and C. But digging deeper, we may see that only protein A can plausibly increase the concentration of this metabolite, and therefore explain the interaction with the environmental factor. And so in this kind of way, we can sort of build this causal diagram where we may explain the genetic effect on the complex trait, on the phenotype of interest, which is in this case mediated by the regulation of protein A and the concentration of the metabolite M, which also mediates the effect of the environmental factor. And this can be mechanistically pretty powerful because it gives us actionable insights. For example, we can then recommend to people that carry the genetic variant G to avoid the exposure to E. Or we can design drugs that lower the concentration of the metabolite M, or target the regulation of protein A, and so on. But importantly, none of these could be conceivable without discovering the interaction effect between gene and environment. And if we look at the known GWAS hits in the phenotype of interest for the PEGS-DREAM challenge in the GWAS catalog and in the FinGen databases, we see that the vast majority of these already known hits would greatly benefit from this further annotation. The second most prevalent class here is intergenic variant, and there are also intron variants and regulatory region variants, which all variants that are classified as simply falling into regulatory regions and which are typically difficult to mechanistically interpret. So in conclusion, we believe that going forward in the future of human population genetics, we should probably think about systematically discovering this gene-environmental interaction, even though logistically in humans this is very difficult. And in the context of the PEGS cohort, by applying the framework we proposed, if we had access to the real data set, is helpful for functionally annotate the already known GWAS hits, but also to identify novel variants that are associated to the trait, but whose effect wouldn't be discoverable if not considering the environmental stratification. And as well as identifying environmental factors, which in some cases we may act upon, whose effect on the phenotype is only discoverable when accounting for the genetic variation in the population. So with that, I just will conclude by thanking our institutes, the European Molecular Biology Laboratory in Heidelberg, Germany, and the European Bioinformatics Institute in England, in Cambridge, and our supervisors, Federico Supervisor Sperborg, and my supervisor Evangelia Petzalaki, who kindly allowed us to come here, even though this has nothing to do with our PhD project. And of course, a big thank to the PEGS Dream Challenge organizers. It's been great work put there, and thanks to you all for your listening.
Thank you, Federico and Alessandro, for participating in the challenge and presenting today. I think it was really interesting to see your work. I want to say that if you are interested in using the actual PEGS data for the second part of the challenge, please feel free to submit a request, and we'd be happy to share the actual data with you for your interesting and novel ideas. But any questions for Federico and Alessandro?
I have a question. So did you try just the polygenic risk score as how was with respect to the, because you know you had how many, 12 PRSs?
Yes.
And one resulting score from the random forest, right?
Yes.
So if you use only the 12, what do you get?
Yes, this is a great question. We did not try that. Simply because for the challenge we had to give a prediction for all of the samples, and the genetic data were available only for about half. So we didn't try that. But I can tell you one individual polygenic scores individually were already ranked by their predictive performance, and the top was 0.736, so much lower than what our model achieved.
Which was in line with I think your baseline. And,
Yeah.
Correct. That was the threshold we set so that people were forced to surpass that and build some interesting models, not just based on the polygenic scores. I think their score was almost 20% improvement over using the polygenic score alone.
I have a related question. PRSs are very dependent on ethnic group. So did you have that into account?
Yes. So this is also why we decided to not rely only on one PRS, but to use this logistic regression, because that would also help us to find which polygenic scores work best and which don't work at all. For example, we had a case. One of the 12 polygenic scores actually had a negative coefficient in our logistic regression. So that was negatively correlated with the hypercholesterolemia, and that we think that might be due to a different population structure in the population where it was trained. But the logistic regression was able to detect this.
Any
Might
other
it be due
questions
to the
for
flick?
Federico and
It could also be
Alessandro?
because
Oh, yeah, go
the
ahead.
was flipped.
Great talk, by the way. Thank you. I just had a question. So if I understood the presentation, and correct me if I'm, incorrect, you're fundamentally looking at, the relationship between alpha and beta 1, I think you call that other variable. Did you look how that deviation changes, like based on input, like if certain environmental factors for some samples cause that correlation to swing one way or the other?
So this is just to be clear, in the second task, it's purely a conceptual exercise because we did not have access to the data. We only had access in the task 1 via submitting, but indirectly, as explained. So that correlation should hold true for the estimates of the coefficients, even in different data sets, as long as the same things have been measured. And I think in the same data set, the true coefficients would be just basically the same, assumed to be just the same, in the absence of interaction. But I mean, there is the original paper that described that statistical procedure. It's a big work, so it goes far beyond that. And I didn't have time to go into details. And in any case, I'm not too expert about it. So it shouldn't depend on the type of environmental factor, and yeah, the correlation should hold true. And this means that you can use summary statistics to discover the interactions with a higher power. Yeah. I just wanted to add something about the performance alone of the polygenic risk scores. I actually thought about running a cross-validation just on the training data, but because it is synthetic, I don't think that would have been. I mean, I actually tried, but it was random. I think it's because it was synthetic. And this is why I asked earlier today if it was possible to still submit some model, because we were very much constrained in what we could do by time. We just did it in free time, but yeah, it would be nice to have still access to the evaluation pipeline to get more insights.
Thank you, Alessandro and Federico.